And with that being said, I'd like to introduce our first trainee speaker, Bhuvan from Queen's University. And today he'll be presenting his work entitled Understanding the Function of CDK8. Take it away, Bhuvan.
Can everyone see my screen? Yeah, so I'm Bhuvan. I'm doing my master's in Arts Physical Lab at Queen's University, and I'm working on understanding the regulation and function of CDK8, and which is a key player in transcription regulation. So transcription is a fundamental process where RNA polymerase synthesis RNA from DNA, and transcription of all genes is not necessary at all times, and a tight control is necessary. Disrupted transcription is often seen in diseases. That is why understanding transcription is important. So CDK8, which is a key player in transcription, is a kinase subunit of this mediator transcription cooperative complex. And the mediator is organized into 4 modules, which is head, middle, tail, and the CDK8 kinase module. So mediator regulates transcription by bridging information from transcription factors to RNA polymerase 2, where the head region interacts with RNA polymerase, where the tail and the middle portion interacts with transcription factors. The CDK8 kinase module, which consists of CDK8, cyclin C, MET12, and MET13, reversibly associates with rest of the mediator complex. So CDK8 regulates transcription by phosphorylating transcription factors and components of transcription machinery. And depending on the protein it phosphorylates, it might lead to activation or inhibition of the target genes. And sometimes it does this by targeting the proteins for proteasomal degradation. So CDK8 is a member of cyclin-dependent kinases. And in general, but unlike other cyclin-dependent kinase, CDK8 does not follow the typical regulatory mechanism. A cyclin-dependent kinase on its own is inactive because a structure called as T-loop or the activation loop is folded and blocks the ATP binding site. In order to become partially active, it needs a cyclin partner, which unfolds the T-loop, making it partially active. But for it to become fully active, phosphorylation of the T-loop is necessary to make it stable. But in case of CDK8, where it binds cyclin C to become partially active, a phosphorylated residue is missing in the T-loop, so we still don't know how it becomes fully active yet. So even though we know a lot about CDK8 and its activity, we don't know how it regulates and how it affects its function. So in order to learn that, I'm going to look into CDK8 regulation from disease-associated variability, and also I'm going to see the effect of phosphorylation on CDK8 function and see how CDK8 MET12 interaction affects CDK8 function. So CDK8 was first identified as an colorectal oncogene, and after that, many cancer studies have reported amplification or presence of missense mutations in cancer. Apart from cancer, many developmental disorders have also reported missense mutations on CDK8. And Sabrina and Jamie, who was an undergrad student in my lab, helped me with making mutants and running Western blots in this project. So in order to identify CDK8 missense mutations, which will likely disrupt CDK8 regulation and function, I use these 2 databases. First is CBioPortal, which contains a repository for sequenced tumor samples. I examined all the available primary cancer studies, where I selected only missense mutations on CDK8, and I focused on CDK mutations occurring on cancers with low mutational burdens. Likewise, I used ClinVar, which is a repository for mutations found on clinical patients, and I selected missense mutations on CDK8. Together, I narrowed down to mutations that fall on identical residues between yeast and human, since I'm making all these mutants in yeast, and which resulted in 23 unique mutations. So these are the 23 mutations which I found from both CBioPortal and ClinVar, and these are the human missense mutations and their identical residues which are found on yeast. when I tried to map these mutations onto the structure of CDK8, I found that most of the mutations were found in and around the activation loop or the T loop, and sometimes on the interaction site between cyclin C and CDK8, or on the interaction between MET12 and CDK8, suggesting that these mutations might affect the activity of CDK8. So to begin with, I selected these 5 mutations. I selected them based on the criteria that they fall in and around the T loop or near the interaction sites between cyclin C and CDK8. And I made these mutations and I tagged them with flag epitope, and I introduced them onto a CDK8 knockout background. And to see if these mutations are producing normal levels of protein when compared to the wild type, I ran a Western blot from the whole cell extract. And as you can see in the CDK8 knockout background containing an empty vector, there is no protein production. But in some of the mutants, I found there is an increase in protein production. To check if increase in protein production is either due to a result of increase in transcription or whether the protein is itself is being stable, I did a qPCR. And as you can see that in the mutants which are having an increase in protein production are showing a clear trend in increase in transcription. So suggesting that increase in transcription is leading to increase in protein production. In order to see if these mutants are growing or showing any growth phenotype, I did a growth assay where the where rows represent tenfold serially diluted E cells, and each column represents different strains. And as you can see, at 30 degrees, which is an optimum condition for yeast growth, all the strains are growing fine. But under Ethereum bromide and galactose condition, which relies on CDK8 function to phosphorylate Gal4, you can see that the wild type is growing fine, but in the absence of CDK8, the knockout is growing sicker. And some of the mutants are exhibiting a knockout-like phenotype, suggesting a disrupt in CDK8 function. Together, it suggests that there is a feedback mechanism where, since there is a disruption in CDK8 function, probably maybe the cells are trying to produce more CDK8. In order to check if the varying levels of CDK8 affects other subunits of the CDK8 kinase module, I checked the levels of MET12, which is also a subunit of CDK8 kinase module, and I did not find any correlation between the varying protein levels between CDK8 and MET12 protein levels. So in the interest of time, I'm moving on to objective 3, where I'm studying how CDK8 MET12 interaction affects CDK8 function in vivo. And if you remember, where I spoke about where CDK8 is missing a phosphorylated residue on the activation loop, so we still don't know how it becomes fully active. This paper, through structural studies, they predicted that the N-terminal region of MET12 interacts with CDK8, which causes a conformational change in the T loop, making it stable, which might compensate for the missing phosphorylated residue on the T loop. So in order to study how these interaction between MET12 and CDK8 affects CDK8 function, I made 2 mutants. First mutant is I449E on CDK8, which they already showed in the same paper that during co-immunoprecipitation, when they pulled down CDK8, they did not observe any MET12 coming down along with it, suggesting that this mutation might weaken the interaction between CDK8 and MET12 in vitro. I also made another mutant with the help of Zhongshan Jia, who is an expert in protein structure analysis, and he predicted that if I make a mutant N48 mutation on MET12 to N48R, it might strengthen the interaction between CDK8 and MET12. So in order to study how this binding strength affects between CDK8 and MET12 affects CDK8 function, I made these 2 mutants. So first I made a MET12 mutant, MET12 N48R mutant, and I tagged with HA epitope, and I introduced them into a MET12 knockout background. And as you can see that it produces normal levels of protein when compared to MET12, and the empty vector does not produce any protein as expected. Likewise, I introduced CDK8 I449E mutant, and I tagged them with flag epitope, and I introduced them into a CDK8 knockout background, and it also produces normal levels of protein when compared to the wild type, and the empty vector does not produce any proteins. So in order to check the growth phenotype, I did a growth assay where again each row represents 10-fold serially diluted yeast cells, and each column represents different strain. And in all the conditions, you can see that both MET12 knockout and CDK8 knockout exhibit same phenotype, suggesting that functional similarity between subunits of the CDK8 kinase module. And in all conditions, the I449E mutant, which weakens the interaction, produces same phenotype as of the wild type. But in contrast, the N48R mutant, which strengthens the interaction between CDK8 and MET12, produces even a sicker phenotype than the knockout in the Ethereum and bromide galactose conditions. Particularly in 37 degrees and 18 degrees, you can see that the N48R mutant produces is sicker in 37 degrees, but it grows better than the wild type in 18 degrees, suggesting this opposing phenotype. And this whole thing suggests that CDK8 and MET12 interaction is necessary, but might be in a condition-specific manner. To summarize, mutants with increased CDK8 mRNA protein levels produced knockout-like growth phenotypes, and CDK8 increase in protein level in the mutants with disrupted CDK8 function suggests a feedback mechanism, and interaction between a CDK8 and MET12 is necessary in a condition-specific manner. I want to thank my supervisor, Dr. Maria Aristizabal, and our collaborators and current and previous members for their constant support and guidance. Thank you.
Great. Thank you so much, Bhuvan, for that really great talk and great use of model organisms to show your work there. So now we'll open up the chat. If anyone has any questions, feel free to put them in the chat or raise your hand and we will unmute you. Go ahead, Ben.
Hi, great talk. I was struck by the phenotype of the mutations how you could, when you strengthen the CDK8 MED12 interaction and you had this phenotype. And I was wondering, is it known if what CDK8 is doing outside a mediator? Like, is this a mediator independent function of the kinase, or what do you think is going on?
Yeah, that's a great question. Actually, we still don't know whether CDK8 performs function on its own with cyclin C, or the whole complex has CDK8, MET12, MET13. They together they perform the function. So yeah, it's still a debate whether we still don't have a clear answer whether the CDK8 performs function on its own.
Great, thanks.
Yeah, I had a question. Does the human CDK8 complement the yeast deletion, or do you need to replace more subunits of mediator before you can get complementation?
I haven't checked human CDK8 whether it complements CDK8 deletion. Here I have made all these mutations on CDK8, yeast CDK8. Yeah, I also don't remember reading any paper where they have introduced human CDK8 into yeast and whether it does any compensatory mechanisms or not. Yeah, I haven't seen any yet.
Thanks.
And we have a question in the chat. On slide 15, why did you choose just those genes?
Just these mutations? Okay.
I think that's what they meant. Yeah.
All right. So yeah, there are 23 mutations. Since I cannot make all the 23 mutations, I focused only on 5, and I selected them based on, since I got all these 23 mutations, whether they're on the T loop, near the T loop, and somewhere on the interaction site between cyclin C and CDK8. So I chose one from which falls under each of those conditions, where I try to cover most of all the 23 mutations as much as I can. But yeah, the criteria was that whether they are falling on or near the T loop or on the interaction sites or somewhere.
Awesome. All right. So with that, I think we're going to move on to our next speaker. So I'd like to introduce Dr. Michael Olson from the Toronto Metropolitan University, and today he's going to be talking about actin cytoskeletal regulation by Rho GTPases and effectors. Thank you, Dr. Olson. We can't hear you. I think you're still muted.
Now I'm unmuted.
Yes, we're good now.
Thanks for unmuting me. Yeah, so I slightly changed the title of my talk as I was putting it together to make it a little bit more specific. It's a story we've been working on it for a few years, and I hope you find it interesting. This is the Toronto Metropolitan downtown campus, so you can see we're really quite downtown. In fact, this building here has my other office, not the office I'm in, but my other office. And there's the CN Tower, everyone knows that. So I want to start with the acknowledgements first, because I always forget to do this later on. This was started by a PhD student I had in my lab, Jennifer Cameron. It was carried on by postdoc David McGarry, and a lot of this work is being carried on by another PhD student, Katerina Majkovic. And we had a lot of collaboration with a mass spectrometrist in Glasgow, Sarah Zanovan. So the question is, why study cell movement? And really, that's what we do. And there's a number of ways that cell movement is involved in numerous biological processes, including embryonic development. This cute kitty is actually a mutant cat. You can see its white belly and white furs is because a defect it has in the way that melanoblasts migrate from the neural crest to the regions where they have to get to, which is why it has these white patches. So it's a black cat. It should be a fully black cat. But because it has defects in cell migration, it has this cute tuxedo look. It's important for how immune cells work. If this YouTube loads, what it shows is a white blood cell that's moving around chasing these bacteria. And again, this is important. This is in vitro demonstration of what happens in our body, where immune cells have to move in order to fight infection. And our particular interest is actually in cancer metastasis. So metastasis involves the movement from primary tumors, either as single cells or cell clusters, through normal tissue into blood and lymphatic vessels, where they then disseminate around the body. And cancer metastasis is really the major problem, really the lethal aspect of cancer. So cell movement is important for normal health and also in disease. And it's really the actin cytoskeleton and how the cytoskeleton dynamically changes, which provides the power for how cells change their shape, how they move, and in the case of cancer, how they invade and metastasize. And cell movement is powered by sort of two things. One is actin polymerization allows cells to protrude. They do this largely in two kinds of structures, in lamellipodium and also in filopodium. So lamellipodium are kind of meshworks of polymerized actin, and filopodium are more linear projections of actin. So as a cell protrudes in one direction, it also needs to bring up the rear end of the cell through actin-myosin-mediated contractile force generation and the retraction of the lagging end of the cell. So you've got these two forces that help the cells move: the actin polymerization moving it forward, and then actin-myosin contraction bringing back the rear. And this is a real picture of an MDMB 231 human breast cancer cell, which kind of shows you in real terms what these structures look like. So this is that meshwork of actin that I was talking about, the lamellipodium. And this is really largely driven by a GTPase of the Rho GTPase family called RAC1. These are filopodia, so these are those actin-rich projections, and they're driven by another GTPase called CDC42. And these long parallel fibers that you can see here are often called stress fibers. And they're regulated by another Rho GTPase called Rho A. So you have the Rho signaling proteins helping to promote these two kinds of actin structures, which together promote cell migration. So one of the questions that we had about the time that my student Jennifer was starting was whether there was a role for reactive oxygen species, which is also abbreviated ROS, in the regulation of cell migration. Hydrogen peroxide is one of the most common forms of ROS. It's a short-lived diffusible second messenger that can be produced in response to a number of different kinds of signaling molecules, and it can regulate protein activity. And in fact, elevated ROS is frequently found in cancer cells, and it's known to contribute to the disease progression. And a number of growth factors and also gene mutations have been shown to both increase cell migration and also to increase cellular ROS levels. So we were wondering what's really the relationship between ROS and cell migration. So this kind of shows you what happens biochemically. This is a reduced thiol. So cysteines have thiols, they have this reduced sulfur, and they're the most common amino acid that is sensitive to oxidation. And when you have something like hydrogen peroxide produced, you can have a mono-oxygenation, which gives you sulfonic acid. And this is quite reversible. This can be easily reduced back to the thiol. But if you have another molecule of hydrogen peroxide, there's kind of three fates that can happen. You can have further oxidation to now one which has two oxygens. This is called sulfonic acid. The sulfonic acids can form disulfide bonds between sulfonic acids, and that's where you get that kind of bridging between proteins or even within domains of protein. And then in terms of experiments, something that one can do is react the sulfonic acid with trapping compounds. And I'll talk about that in a bit more, which is a way that we can really monitor the redox state of the proteins. And finally, it's also possible for another hydrogen peroxide to promote the further oxidation to sulfonic acid. Sulfonic and sulfonic acid are not readily reversible. So that when they get to this point, they tend to stay in that form. I mean, it's not completely reversible, but it's energetically more difficult, whereas this is quite easily to reverse in both directions, either the oxidized or reduced form. So the question we had is, can we actually detect hydrogen peroxide in living migrating cells? And this was enabled by the publication of a probe called HYPER. It's a fluorescent protein that can be used to sense hydrogen peroxide in live cells. In this case, what was done is that yellow fluorescent protein was inserted into the regulatory domains of the bacterial transcription factor OxyR. And in the presence of hydrogen peroxide, this forms the disulfide bonds between two parts of OxyR. That then changes how the fluorescence properties of the YFP. And so this schematic diagram gives you an idea. Here is the YFP between these two domains of OxyR. Here are the two reduced cysteines. In the presence of hydrogen peroxide, you get that disulfide bond formation. And this brings the two domains together. And this causes a conformational change in YFP. And then, like I said, it changes the fluorescence properties of the YFP. And this can be sensed in terms of looking at a shift in the excitation of YFP. So it goes from a normal excitation emission properties. So normally it's excited at 405 nanometers, and it's better excited at 488 nanometers. And then emission is at 560 nanometers. So there's a change in these excitation emission properties, which can be monitored. But we also reasoned that this would also affect other properties of the yellow fluorescent protein, including fluorescence lifetime properties. And we were well set up to do fluorescence lifetime imaging microscopy. So that's actually what we did to monitor and to sense or to locate where hydrogen peroxide was being generated. So this film, which should play, it should load. Okay. It's a cell expressing HYPER, and it's been given 2 hydrogen peroxide doses into the medium. And what you could see is that this thermometer here is basically showing fluorescence lifetimes, and the warmer colors you saw were shifted to the colder colors by those 2 hydrogen peroxide treatments. And this is what it looks like in terms of quantifying this. So the hydrogen peroxide comes here. There's a drop in the fluorescence lifetime. Hydrogen peroxide is quite short-lived, and so over a few minutes, it basically is degraded, and then the disulfide bond in the YFP or in the OxyR YFP is reduced, and the fluorescence properties of YFP go back to normal, and then it can be stimulated with hydrogen peroxide again, and we can see the same kind of drop in the lifetime. So what this shows, just by adding hydrogen peroxide to media, is that we can sense this. It's a reversible process, and so this makes a nice probe for visualizing hydrogen peroxide in living cells. So the migration model that we often use, these are DMB231 cells. One of the things we do is create a scratch in a monolayer and look for cells moving into that gap as a way of stimulating their migration. And this is, again, looking at the hypercyto. So this is cytoplasmic localized hyper and the lifetime map. And this is untreated and this hydrogen peroxide treated. So again, you can see the slightly warmer colors get colder, they get more blue. So that's indicative of this drop in fluorescence lifetime. And so that's by using hydrogen peroxide. And then when we just compare cells that were stationary versus those that are migrating, it's not a huge drop in absolute terms, but it's a significant drop in the migrating cells, which indicates there is indeed a significant there are significantly higher levels of hydrogen peroxide in the migrating cells relative to stationary cells. We made another probe where we actually anchored the hyper to the plasma membrane because we had an idea that maybe most of the hydrogen peroxide generation was actually happening at the plasma membrane. You can see again, hydrogen peroxide causes a shift in the color from slightly warmer to colder. And it's a similar thing that comparing stationary to migrating cells, again, there is significantly more hydrogen peroxide in migrating cells right at the plasma membrane. But one of the things that my student, Jennifer, noticed, which was really noticeable, was that when she just looked at within single migrating cells, there was an obvious difference between the amount of hydrogen peroxide at the protrusions relative to the cell body. So if you look at these two protrusions that are slightly more magnified, you can see they're bluer than here in the middle of the cell, which is slightly yellower and redder. And looking at this from a number of cells, what we see, in fact, there are higher levels of hydrogen peroxide in the protrusions compared to the cell body in migrating cells. So migrating cells produce more hydrogen peroxide, and it's actually quite localized to protrusions. So are ROS necessary for cell migration? The thing my student did was to track the migration of individual cells in the scratch wound assays, and then to add different amounts of a ROS scavenger called N-acetylcysteine. So they provide basically a reduced thiol that can be oxidized and scavenge any hydrogen peroxide that's being produced. And you can see there's a dose-dependent drop in the migration of the cells. So this ROS generation actually helps promote the migration of these cells. And this is just showing that these are significant effects at these different N-acetylcysteine concentrations. So hydrogen peroxide is being produced, it's necessary for migration, but this is really leading to protein oxidation. And this is what I said before, this is a similar sort of schematic. Here's the thiol, reactive oxygen is generated, it can lead to disulfide bond formation, lead to further oxidation. But we can also trap it with molecules like dimidone, and this causes a covalent reaction with the sulfenic acid. And this cannot be reduced, and this dimidone can be detected by mass spectrometry or even with an antibody against the dimidone moiety. And so what we did is we took confluent layers of monolayers of dishes of cells. In order to induce migration, what we did was scratch off about 2/3 of the cells on the dish and then wait about 2 or 3 hours for them to start moving, and compared the amount of protein oxidation in stationary cells versus migrating cells. And what we can in fact see is that there's more protein oxidation that you see in migrating cells. And by mass spectrometry, what we identified was a few hundred proteins. The bulk of them could be classified as being involved in cell communication. A lot of them were involved in the cytoskeleton, and a lot were involved in metabolism. So there's a whole variety of proteins that are being oxidized. But we were very interested in these cytoskeleton proteins because, as I said, cytoskeleton is very important for cell migration. And we really focus on a particular protein called cofilin-1. Cofilin has 2 cysteines that are quite exposed to the solvent. And what this model shows, the sulfurs are in yellow, and these 2 red balls are 2 oxygens that have been reacted with that sulfur. And so this is where we modeled that. And then again, based on modeling, where cofilin had been placed in contact with actin, what we can see is that these 2 sulfurs are actually very close to that interface with actin. So what we believe, again, this is modeling, but what we believe is happening is that 2 things happen. The addition of the oxygens causes a sort of a steric clash with the surface of the actin. And also the negative charges can then cause charge-charge repulsion, where you've got negative charge on the actin. So between these 2 things, we think that this lessens the interaction of cofilin with actin. And what we found, in fact, was that the 2 cysteines, 139, 147, were sensitive to oxidation, and this led to reduced binding of globular actin, reduced binding of filamentous actin, and also an inhibition of the cofilin's normal activity, which is filamentous actin severing. So because the cofilin gets oxidized, it can't bind F-actin, it can't sever F-actin, and this promotes filamentous actin stabilization. So is there a role of cofilin oxidation during migration? What we did is we expressed fusion proteins of cofilin, which is fused to the M-cherry fluorophore. So this is M-cherry, and this is the M-cherry cofilin fusion protein. And we either had wild-type cofilin, or we changed those 2 cysteines to alanines. And then we looked at the effect of introducing either more wild-type cofilin or non-oxidizable, and therefore non-turn-offable version of cofilin. And this is a kind of slow movie, so I'll kind of accelerate this, but we looked at single-cell migration. What we found is that the wild-type cells, when they're plated, they spread more quickly, they polarize more quickly, and they start to migrate more quickly. As I said, this is a bit slow, so I'm going to go cut to the chase. These are spider plots that show you tracks of individual cells. So what we can see is that compared to cofilin, those ones that are expressing the non-turn-offable cofilin don't migrate as well. And in fact, what they do is they don't migrate in a directional manner. What they do tend to do is just kind of turn back and forth on themselves. So they don't make forward progress. So they drop in this directionality parameter. As I said, anecdotally, we could see that they spread less well. So we used a machine called an excelligence to look at a parameter called cell index and found that the mutant cofilin cells do in fact spread less well. And that's what this shows. And then finally, we measured their force of adhesion, and the ones that are expressing the cofilin that can't be turned off are actually less adherent to the substrate. So this is the model we came up with. This is a polarized cell. This is the zooming in on that. Here in purple is a gradient of some kind of extracellular chemoattractant or hydrogen peroxide itself. This leads to a gradient of intracellular hydrogen peroxide being produced. At the front of the cell, the protrusions, cofilin is being oxidized. It doesn't bind F-actin, therefore it doesn't sever F-actin, and this helps the lamellipodium push forward that network of polymerized actin. But further away, where there is less hydrogen peroxide, cofilin tends to be more active, and it tends to then sever the filamentous actin and help return G-actin to the front of the cell, where it can be used for new actin polymerization. This is a model, but it's consistent with our observations. So that was all a nice study, but we started to ask some questions. What are the enzymes that might be producing the reactive oxygen species? And so we looked by RNA sequencing data we had from the 231 cell line. What we found is that a lot of the enzymes that are known to produce hydrogen peroxide of the NADPH oxidase family are actually expressed at relatively low levels. But another protein called MyCal-1 was actually expressed higher than any of those NADPH oxidase type proteins. So we got quite interested in MyCal-1. So it's a protein that has these domains, and it's a monooxygenase domain. There's a calpona homology domain, a limb domain, and a binding domain. What it does is it takes NADPH and then produces hydrogen peroxide from oxygen. Oxygen actually directly oxidizes actin on 2-methionine residues, and that causes the depolymerization of F-actin. So MyCal-1 produces hydrogen peroxide, and that's what one of the things it can do is actually directly depolymerize F-actin. So we CRISPRed it out with a few different sgRNAs. So now we have 231 cells where we've knocked out MyCal-1, and we can ask questions about what is the purpose of MyCal-1. And one of the things that we noticed is that we see sort of an emergence of these longer actin filaments, which is consistent with MyCal-1 being gone and actin filaments being stabilized. And we also noticed that cells are actually more spread and actually bigger. So we see that they occupy a bigger area on the plate. And this F-actin anisotropy is a measurement of the actin organization. And why these values being higher is really an indication that there's more parallel fibers, which is what we see. When we compare the scratch wound assay, which I introduced earlier, we can see that within about 36 hours, cells that have a non-targeting control can completely close these gaps, whereas the two different knockouts we used are impaired in their ability to close the gaps, indicating they are less good at migrating. And this just shows that quantitatively, it's a significant effect. When we look at random cell migration, we did this by, in this case, using automated cell tracking. You can see the paths of a number of different cells being highlighted here. And then this animation shows you these in a spider plot. So these are the non-targeting controls. And at the same time, this is the migration of MyCal-1 knockouts. So you can see these ones are migrating away from that sort of 0-0 coordinate origin better than the knockout. So there is a migration defect, which is manifested both in single cell migration and in scratch wound assays. And this, again, is just quantifying that from a number of experiments. So this looks at these track lengths. So there is a significant effect on the ability of these cells to migrate. And this is, again, is reflected in the fact that they tend to turn more often. So they're less good at moving in a directional way, which is similar to what we found when we're looking at cofilin overexpression. So then I sort of introduced in the beginning that we were interested in the Rho GTPase signaling family, and we wanted to see, is there a connection between Rho GTPases and the regulation of the mycoprotein? Myco is directly sort of having a direct effect on actin. So we wonder, is this a way that you can, in addition to what's already been shown, to connect Rho GTPase signaling and myco-1 regulation? So myco-1 has been shown to be a phosphoprotein. You can look on phosphocyte, and there were loads of phosphorylations reported. So it seemed kind of likely that maybe kinases were the link between the Rho GTPases and the mycos. So my postdoc David did this experiment. It's a little bit complicated to explain, but he expressed two different active forms of either Rho or CDC42 alone, or with their effector kinases that they bind and regulate. PAC1 and MRCK alpha are effectors of CDC42. ROC1 and ROC2 are effectors of the Rho GTPase. And so this just shows the GTPases being expressed. Here are the ROCs and MRCK up here. Here's PAC being expressed all in green. And here in red is myco-1. This is a flag tag version of myco-1. And what he then did was to immunoprecipitate with an anti-MYC antibody, the green proteins, and look to see whether red flag tag myco-1 would co-immunoprecipitate with any of these proteins. And the only one that was really significant was this PAC1 plus CDC42. So this is saying is that there's a stable enough interaction between PAC and myco-1 that you can co-immunoprecipitate it, which was very interesting because now maybe we have a direct relationship or direct path from Rho signaling to myco-1 through this kinase. So this is an experiment looking at actin in vitro. And so this is globular actin and this is F-actin. And what David did was to use high-speed ultracentrifugation to either pellet or leave the actin in the supernatant. So globular actin stays in the supernatant. You can see here it's all the supernatant fraction. But if it's being polymerized in vitro to F-actin, most of it is found in the pellet fraction and less of it is found in the supernatant fraction. So we can look at the state of the F-actin G-actin relationship by using centrifugation. When he added PAC1, it doesn't do anything on its own. When he added myco-1, now we're starting to see some actin depolymerization, so there's a shift more towards the supernatant fraction. But when he added F-actin plus PAC1 plus myco-1, now we see a big shift. So now it's all the supernatant fraction. What this is saying then is that this PAC1 association with myco-1 is increasing the ability of myco-1 to depolymerize F-actin. And that's shown more directly in this fluorescence assay of actin depolymerization. So in this case, actin's been conjugated with a fluorophore called pyrene. It has a certain level of fluorescence when it's incorporated. And if it gets depolymerized into G-actin, the fluorescence drops. So what we see here in the control of the F-actin, we have a sort of fairly stable fluorescence signal. You add PAC1, makes no difference. Add myco-1, there's a certain drop in the fluorescence, indicating there's depolymerization. Add myco plus PAC, the drop is bigger. So that agrees with this experiment up here. And this just quantifies the number of experiments. We can see PAC doesn't do anything. Myco does something significant. That combination of myco plus PAC does the most. So the PAC is accelerating myco's ability to depolymerize F-actin. So we mapped two phosphorylation sites that PAC is doing on PAC1. Here at serine 817 and serine 960. And in the three-dimensional structure that's been predicted by AlphaFold, 960 is here at this turn between these three alpha helices, and 817 is here at this particular alpha helix. This is the monooxygenase domain. This is the Calpona homology domain. This is the sort of Rab binding domain. And it's been shown before that these regions basically keep the protein in a closed conformation, and they have to be shifted to allow the monooxygenase domain to be activated. This can be done by Rab binding to the Rab binding domain. And what we're saying is that we think that the PAC binding and phosphorylation of this site similarly shifts these alpha helices to increase the myco-1 activity. So this is our final model. This incorporates some other experiments I didn't show. So PAC1 exists normally in an inactive dimer. CDC42 is here. And as I said, Michael-1 exists in a closed, inactive conformation, so it isn't doing anything to F-actin. What we found is that certain growth hormones like basic FGF will bind to its receptor, activate CDC42, bind to PAC, increase PAC autophosphorylation. So now you've got this CDC42 PAC in an open conformation that then binds to Michael-1 within the monooxygenase domain. You get the phosphorylation of these two sites, serine 960 and 817. This increases the activity, so actin is now depolymerized. And at the same time, the opening up of the protein also allows for the binding of other proteins like RAB10 to the RAB binding domain. So it helps to open up the protein and enable it to act as an even more of a signal integrator between the Rho signaling pathway through CDC42 and the RAB GTPase signaling pathway. And this happens in response to growth factor receptor stimulation. So FGF2 initiates this cascade and leads to F-actin depolymerization. So that's the end of my slides. I want to thank the people who funded this, the funding from CIHR, Canada Research Chair to me, and Circus funded this, and we also had some money from the Ontario Institute of Cancer Research. And I'm happy to answer questions.
Some very, very great work over there. So if anyone has any questions for Dr. Olson, feel free to drop them in the chat or raise your hand. Go ahead, Matt.
I can get started with the first question. It's more just kind of an interest question, and yeah, I don't know. Why did you guys make alanine mutations in the cofilin 1? Like, I assume cysteine is often mutated to serine, right? So I'm just curious about the rationale for alanine.
That's a very good point, and that was largely based out of ignorance. So I kind of told my student to change them to alanines because I'm just used to coming more from a kinase field. You know, sort of default mutations that people make of serines and threonines is to alanine. So I just told her, change it to alanine. And, you know, then in retrospect, it would have been more consistent with what people do to change the cysteine to serine. So I was a bit worried about when we sent the paper off, whether reviewers were really going to be difficult about the fact that we hadn't done the traditional mutations, and there was no mention.
Okay.
So we kind of, I don't think it would make any difference. I think if we change cysteine to serine, I think we get the same results. But yeah, I mean, I'm pretty sure we would get the same result, but we kind of got away with it.
Yeah, the data looks convincing. I was just curious. Thank you.
It wasn't for any sort of intelligent reason.
All right. Stephen, go ahead.
Can you start my video?
We can hear you. We can hear you. Yeah.
Okay, all right. Be faceless. So Mike, great talk. I enjoyed that. So a couple of questions. How come you think it's CDC42 and not RAC1, which can also bind to PAC and activate it, as you know?
That's a good question. So I kind of, slightly obfuscated the history of this project. And what we'd actually done was we had done a bioID screen with Michael-1. And we found PAC and we found CDC42. We didn't find RAC. And so we kind of used that to guide how we did that experiment. The way I presented and the way we put it in the paper was that it was like we had logically come up with this hypothesis that maybe these proteins were interacting. But we kind of reverse engineered back from the bioID data. And then we never, it was a thing that I always wanted my postdoc David to do, to go back and look at RAC in parallel to see whether we would get the same effects that we'd seen with CDC42. And he never went back and looked. So it's an open question. My prediction is it would be similar, but until it's really tested, we can't know that. Right, because I mean, RAC would be the one that's at the so you don't show it in tiny little filopodia, but it's at the front of the migrating cell, right? More in the lamellipodial kind of region, right? I agree. You could argue it would be more consistent with the overall story, right, that I told. And like I say, it's something. It's you get on a certain track and you do certain experiments and you have ideas what you should go back and do, and they don't always go back and get done. And I feel like we should go back and do it, but I'm not sure we will.
Yeah, because you don't show, or maybe you haven't shown it, but do you have any data showing that either CDC42 or PAC binds directly to MyCal1?
Oh, I think Dr. Olson lost connection, Wi-Fi, potentially.
Yes, didn't like
We're
my
just gonna give
question.
him a minute, if that's okay.
Okay, scared him off.
Okay. Well, I'm glad that didn't happen during the talk, at least.
Yeah.
Okay. Well, I'm gonna give him another couple minutes. Sorry for the interruption, everyone.
It's all right.
I think we got him back. Just joining.
Hi, I'm back. Sorry, my Zoom crashed.
No worries. I think Stephen had another question. Oh, Stephen, go ahead. You're muted now.
Okay, yeah, all right, thank you. What did I say? I think I was asking if you had anything data showing that CDC42 or PAC directly binds to MyCal1. And I thought maybe the RAB binding domain might be also a Rho binding domain kind of thing.
Yeah, it's a good question. So we actually did domain mapping and, deleted the different components of MyCal1. Where we found PAC was binding was predominantly to the MO domain and better to the MO and component homology domain. And that's the region that Actin binds to. So those domains form a kind of a cleft. And we think that the PAC fits into a similar cleft that Actin does. So yeah, it's not binding to the RAB binding domain. And we did some other experiments that showed that the CD42 is not binding directly to MyCal1. That's just part of a complex with PAC.
Okay, cool. Maybe we could talk more at another time.
Sure. I'm easy to find my email is easy to find.
Okay, cool.
Awesome. And we have a question from Donovan in the chat. Great talk. Do you think the local environment is as important as the intracellular H2O2 in your model? Or is the cell able to sustain this movement independent of environmental hydrogen peroxide?
That's a good question. And I have to admit, we went with a certain idea that the hydrogen peroxide. Am I frozen again? Oh, we went with a certain idea with that it was intracellular hydrogen peroxide that was important just because of having done the imaging. And, what we saw with hydrogen peroxide in the migrating cells and, at the plasma membrane, at the protrusions. And in that case, we weren't adding external hydrogen peroxide. So we've really focused on the internal and the sort of self-generated hydrogen peroxide. But undoubtedly that, there are sources of hydrogen peroxide like neutrophil, the neutrophil burst, will release reactive oxygen species. And it could also be that there's external environmental hydrogen peroxide could also influence migration. We really not looked.
All right. Vincent, go ahead.
Can you hear me? I'm not sure.
Yes.
I'm not usually working from this station. Okay. I don't know much. So I'm naive to this field. And I'm wondering in what biological context do you would it make sense that a gradient in hydrogen peroxide would drive cell migration? Like say would it be in wound healing or some developmental process? You mentioned cancer in your introduction, so what are your thoughts?
Yeah, it's a good question. So, and we're doing this kind of thing, which kind of starts from a biochemical level, and you try to build out and make it relevant to the whole cell. We were able to put together a pathway that goes from growth factor receptors through to PAC activation, through to microphosphorylation. So we do think that there's probably a role in, extracellular or external stimulation through things like growth factors to turn this pathway on to influence how cells migrate. So there's that in terms of sort of physiological relevance. Whether it be involved in something like wound healing, possibly. And then in looking for where it may be involved in cancer, one of the things that is quite striking is that there's an organization which is run out of I think it's the Sloan Kettering Cancer Institute and the Broad Institute, looking specifically at metastatic breast cancer and then doing transcriptomic, genomic analysis in that particular patient population, just metastatic breast cancer patients. And what they find is that there's MAC1 amplifications in something like 40% or 40% plus of patients with metastatic breast cancer. So it's not proof, but this is a very interesting association that there may be a role for elevated MAC expression as a promoter of breast cancer metastasis. So it's something we're actually quite interested in exploring further. But it's not proof. It's a tantalizing lead that there may be just like a basal contribution that you need these proteins to be there to efficiently migrate, but you may be able to actually promote migration by increasing their activity or expression.
Thank you.
Thanks.
Bruce. Bruce, we can't hear you.
That's odd. The microphone is.
You're good now.
Okay, now we can. Now we can.
Sorry, it's just a very slow mouth click. I was just curious as to, so peroxide is also a strong activator of the p38 MAP kinase pathway, and one of the terminal proteins of that pathway is HSP27, which is an actin capping protein. So I was wondering if that pathway would also fit into sort of coordinated cell activity, such as migration, directed migration, versus just some sort of a random, well, less ordered event like restructuring the extracellular, the cytoskeleton itself.
Yeah, no, that's a very good point. So going back to the original work that we'd done, where we were looking at stationary cells versus migrating cells, and then I pulsed them with dimidone to label proteins that had the sulfonic acid, and using mass spectrometry to identify labeled proteins. The biggest group of proteins we got were actually those involved in intracellular signaling. So I do believe that there could be a role in affecting signal transduction. I can't remember if P38 was one of the proteins that we found or not. I don't, off the top of my head, don't remember if we found HSP27. But it's not just cofilin. It was one of these things. Cofilin was quite a major hit in the mass spectrometry. Cofilin is a very abundant protein. It's a very soluble protein. It flies very well in mass spectrometry. So it may have been a good hit just because of, things that aren't necessarily because of its biological prominence. It was a very good hit in the mass spectrometry, and we kind of followed that, and we had the reagents, etc. And then again, never went back and drilled down in the list of all those other proteins that we identified as being increased in their oxidation. But I'm pretty sure there are more stories that could be made from another of other pathways and cytoskeleton proteins that we've just never gone back and really investigated.
Cool, thanks.
All right, and I think this will be our last question from Michael. You're unmuted.
Hi, thank you very much for the talk. I enjoyed it. I was wondering, I know, the mitochondria can secrete a lot of or produce some H2O2, but it sounds like this protein, the Michael one you're discussing, probably is producing much more H2O2 in the cytosol. But I wonder if you've tracked mitochondria, if they are accumulating in the pseudopod, or you don't think that's what's happening.
Yeah, it's a good question. It's something that I've always been kind of concerned about, to tell you the truth. Like, how do we know that, the proteins we're talking about are really proportionally important sources of ROS? It was part of the reason that we built the hyperfluorophore that was anchored to the plasma membrane, to try to, like, to some extent, physically distance it from the mitochondria, so we could see, are we really detecting elevated hydrogen peroxide at the plasma membrane, close to the cortical actin cytoskeleton, and hopefully away from the bulk of the mitochondria. Again, it's not proof, but it's consistent with, the idea that there are other sources of ROS that are, appear to be contributing to the regulation of the cytoskeleton. But it's impossible to, exclude the possibility that mitochondria are also contributing, because as cells move, they actually burn more energy, and mitochondrial activity is probably higher to make up for the fact, and they're probably generating more hydrogen peroxide and other ROS from the mitochondria. It would be not necessarily a byproduct, but a less direct way of producing the ROS, where I think, the Michael enzymes and the NADPH oxidases are regulated, localized proteins that are doing a certain function in the cell, producing hydrogen peroxide in certain parts of the cell in response to certain sorts of stimuli. So it's kind of, you could call it a more regulated process, whereas the mitochondria, it's just pumping out ROS as a byproduct of metabolism, and it's not really the same thing. This is very hand wavy, but
No.
I agree, we can't really exclude that mitochondria may still be playing a role.
I'm not sure if I've missed it, but were you able to localize the Michael enzyme? Where is it localized to?
So it's not, it's fairly much throughout the cytoplasm. The one thing that we did do in that publication is we did proximity ligation to show where PAC was interacting with Michael, right? So we were just looking specifically for that complex, and we found it fairly evenly distributed throughout the cytoplasm. It wasn't concentrated at the plasma membrane. It wasn't anywhere in particular. It was fairly evenly distributed. So yeah, and then we didn't look at it at the same time. We didn't look at where the mitochondria were, so I can't really tell you whether it's near high areas of high concentration of mitochondria are excluded from those areas. I can't really comment.
Yeah, thank you.
All right, thank you so much for everyone's questions, and once again, thank you so much to our speakers for some really lovely talks and some really interesting science. Give them one more round of applause. Before we go, I also wanted to remind you all that we will be having another seminar in December, so feel free to join us for another round of talks. Alrighty, have a great day, everyone.
Bye.